Hello, good morning, and welcome to the 10th series of the International Plant Breeding Seminar, hosted from NC State in Raleigh, North Carolina. Here is the calendar for this 10th series. We have a very interesting topics that you will be hearing during the course of the next 6 Thursday, ending on November 20th. Today, we have the pleasure to have Dr. Rohayu Maharup from the University Malaysia Terengganu. Dr. Rohayu focuses on research in areas of genetic diversity, physiology, and applications of different techniques in plant breeding. She's focused on improving crops for tolerance to abiotic and biotic constraints. She has worked a lot in rice, but also worked in cucurbits. But her presentation today will focus on the pre-breeding in vanilla. Rohayu did her graduate studies in Sydney, Australia. And Rohayu, it's a pleasure to have you with us. Let me stop sharing here so you can share your screen, start your presentation.
Good morning, everyone. I'm Rohayu from UMT Malaysia. Really appreciate to be here, and I would like to share our work on pre-breeding in vanilla. Okay, here the outline that I will go throughout this presentation. A bit of introduction. The orchid genus vanilla belongs to the Orchidaceae family and is viewed as the world's costly spice close to saffron. The vanilla orchid is a climbing vine with green stem and arrow roots. It produces pale green-yellow flower and long pods that mature into commercially valuable vanilla beans. There are 110 species of vanilla orchid, but only few species of vanilla can produce the vanillin for commercial production. USA is the major consumer of vanilla, followed by Europe and Japan with total value of $951 million. It needs warm temperature, humid and 50% sunlight. 600 meters above sea level is encouraged and tends to favor its cultivation. Flowering terrestrial and climbing plant requires 3 to 4 years to set flowering and afterwards flower once a year. The vanilla bean is allowed to develop for 8 to 10 months before harvesting. Vanilla flavor was used extensively in food products, especially in non-carbonate and carbonate beverages. Vanillin also have an anti-cancer and antimicrobial properties. Worldwide total demand for vanillin is more than 10,000 tons per year, of which is less than 0.5% made out by vanillin isolated from vanilla beans. The price is quite hard. As you can see here, is between US 1,200 per kilo and US 4,000 per kilo, while synthetic is less than US 15 per kilo. Range of product includes vanilla extract, powder, premium vanilla infused coffee, vanilla pods, keto sweeteners, massage oil, hand sanitizer, mosquito repellent, and perfume. These products are already available in the Malaysian market. In Malaysia, we only have 2 varieties of vanilla, which are Vanilla Planfra Andrew, mostly cultivated, and Vanilla Tahentensis, Jablumor, usually cultivated in Borneo, in Malaysia. Therefore, we have less vanilla varieties option for farmers. At this moment, no gamma radiation has been exposed to vanilla cuttings for new variety development. So we start this research in end of 2017. So since we have lack of variation in the gene pool of Vanilla planifolia, where only 3 species of vanilla are grown commercially because of their aroma like Vanilla tahentensis, Vanilla pompona shide, and Vanilla planifera andrew, only 2 species of vanilla are important in terms of commons and cultivation that are Vanilla planifera andrew and Vanilla tahentensis. Therefore, by creating mutation by gamma irradiation, vanilla breeders may hasten the development of new varieties with improved qualities, increase the genetic diversity required for long-term crop resilience. Here are mutation breeding. Cuttings of vanilla Penampilan Andrew were collected from industry, Amani Vanilla Temerloh, and then the cutting we take with 3 notes each. Then harvested from healthy vines. Then the cutting were damped with moist tissue paper in plastic bag in an ice box filled with an ice pack to maintain the freshness. And then we place the cutting into the tank inside the acute radiation facility, which is a cesium-137. And the vanilla cutting were irradiated with a 4 dose of gamma radiation, 10, 30, 40, and 50 gray. Okay, irradiated cutting were pre-treated by soaking in wood vinegar. Okay, here you can see the cuttings were soaked with the wheat vinegar in 5 ml in 1 liter of tap water before cultivation to prevent rot and other infections. Then the cutting were cultivated in the greenhouse condition. After the treatment, what impacts on the mutated vanilla Penampilan Andrews plant at first, second, third, and fourth vegetative generations? Let's have a look. Yeah. First is the protein differential. Let's look what happening in first generation. We determine high protein expression in vanilla elite cultivar and the improved mutant lines by using SDS pitch. Based on the previous research, it is more reliable tool and accurate technique to discriminate between cultivars, low cost and relatively simple technique that has been used effectively with no deciphering genetic diversity among or between genotypes in different plant species. So the dose of gamma rays at 10 gray gave the highest number of protein fragments which detected polymorphism between the control, here is the control, and the plants treated. The protein was separated according to the relative molecular size here. And then the marker showed the highest polymorphism percentage compared to random amplified DNA RAPD and in ISSR marker. We also look at the protein concentration. So the result revealed the highest protein concentration in 10 gray for the protein concentration compared to different treatments in M1V1 leaves. From this result, 10 gray or lowest dose of gamma radiation shows the highest protein concentration across all the cultivar. Okay, vanillin, the principal component of vanilla extract, was further quantified with HPLC. Vanillin concentration was significantly affected by the treatment in M1V1 with 10 gray enhancing the amount of vanillin present in the leaves. Ginseng radiated with gamma rays in vitro yielded a higher level of ginsenoside. Similarly, crucial secondary metabolites were reported to increase significantly owing to the treatment with gamma radiation. The result here is the second generation. It showed that the growth development of second generation plants at 0 gray is highest compared with other dose of gamma radiation in terms of shoot measurement till 24th week of cultivation. The least active growth development was obtained from 40 gray. Pigments in second generation. What we found in treated plants has significantly less than control in pigments. The reduction in photosynthesis pigment could result from the effect of the ionizing radiation treatment, as they have been noted to have a greater influence on succulent living tissue. And the degrees of reduction could be of varying extent in different plants. Let's look at the third generation. What is happening to the photosynthetic pigments? We found that the third generation possess photosynthetic pigment that were significantly lower only in 30 gray, while 10 gray show no significant difference with the control, and the pigment ratio show no significant difference at all regardless of the treatment dosage. This observation could reflect the dose-dependent damage caused in first generation, which may be milder in 10 gray treated cutting than in 30 gray treated cutting. This indicates that the treated plants are approaching stability and healing from the impact of the ionizing radiation treatment. Let's look at the fourth generation of the pigments result. The enhancement on pigments was observed to have been improved starting from V3, the third generation, and it's clearly seen in fourth plant generation. Ameliorative effect was well expressed in treated fourth plant population as they were significantly higher than the control in photosynthetic pigment and pigment ratio. The enhancement in photosynthetic pigment in the latter generation, fourth generation, reflect the desirable effect of the gamma radiation and the stability of the plant. Based on previous research, application of 30 gray and lesser dose of gamma significantly elevates the photosynthetic pigment content of lettuce seeds as well. Let's look at the total phenol content on fourth generation. Similarly, the total phenol content was significantly improved by the 30 gray treatment in fourth generation. The observed effect of gamma radiation of the physiology of plants could be because of the generation of free radicals from water hydrolysis, which could facilitate thylakoid membrane dilation. Changes in photosynthesis regulate the antioxidant system, build-up of phenolic compounds, and photosynthetic pigments. Gamma ray could affect changes in plant tissue and metabolism through dilation of thylakoid membranes, alteration in photosynthesis, antioxidative structure modification, and secondary metabolite accumulation. This observation aligns with recent reports where gamma radiation significantly enhanced the total phenol content in barley and wheat. So, the photosynthetic pigments and secondary metabolites, what is the link between them? So, the biosynthesis and production of catechins are facilitated by the enzyme of the phenylpropanoid pathway. And the vanillin and its derivative have been linked to phenylpropanoid pathway by several workers. For example, in tomatoes, Solanum lycopsychum mutans producing desirable secondary metabolites in both the fruit and leaf were reported to possess a higher photosynthetic pigment. This support the deduction that there is a positive relationship between photosynthetic pigment contents and valuable secondary metabolites in mutant plants. Now, we accept inbound students from Indonesia to learn and attach with our postgraduate students. We also have organized community and single mom to learn about vanilla cultivation. Then we're also planning to collaborate with post-harvest and food technology department team for vanilla-based product and cafe. Here, our fourth generation is ready to do for the field trial in fifth generation. Currently, we are implementing different kinds of stress. Here you can see the vine is under stress-induced flowering. We are implementing different kinds of stress on our matured vanilla plant and roots and mutated plants for flowering induction. Hope we can meet again in future to talk about this study. In summary, greater polymorphism reflects higher genetic diversity and first generation from cuttings exposed to 10 and 30 degree dose of gamma rays that have shown polymorphism compared to the control in their protein profiles. High TPC and vanillin content in our mutant lines, it may be contribute to the new variety with significant high value product and may increase the price compared to other variety. However, further observational data is needed to measure and record until 5th generation to ensure the stability of the mutated traits. Feel free to email me if you have other questions regarding to this talk, or you may interested to collaborate with us, or you may want to do inbound student exchange. Just for information, usually in UMT, for those who did inbound student exchange will need to pay bench fees depending on project and faculty. And sometimes also could be free if you are not attached with the project. It's just you are collaborate with other final year student and just join for few weeks. Okay. My profound gratitude goes to Almighty Allah who has been sustaining us till now and beyond. I thank my students, Mr. Basit, Ms. Huma, Ms. Turga, Mr. Muhammad Isa, and Ms. Nur Shazwani for their efforts towards helping Vanilla Research a success. We also get the internal grant from UMT, TAPE RG, for this research. To announce the latest release of our book, Current Agriculture and Farming Technology in Malaysia, by end of this year. Okay, this book about the agricultural sector in Malaysia, where tradition meets transformation and innovation, heralds a new era of productivity and sustainability. I have ordered few copies for myself. And are planning to give away 7 books for special price to 7 different countries, just for USD 20, include postage. I will pack nicely and post it to you, to your address. Okay, please email me by end of this year to ask for copy if you want to buy through online. Okay, feel free to visit our Penerbit UMT link. Here's the link. Okay. Okay. That's it for tonight. Okay. Thank you for your attention. Nice to meet you and see you again.